Okay, now student. So this is, this is topic number 6, and the title of this topic, as you can see, the plant nutrition. Okay, because if you are going to study the syllabus, in syllabus, the topic number, 6 is plant nutrition. Okay, so we are going to just try to complete this topic. Okay, so I'm going to start the performance slideshow. Okay, start from the beginning. Yeah. Now you can see here, this is plant nutrition. Plant nutrition means we have to study the photosynthesis. Do you have some idea about photosynthesis, Imas? Hello?
I thought I was not on mute.
Okay. I'm asking you the question. Do you have some idea about photosynthesis?
Yes, I do.
Okay, okay. What is photosynthesis? Can you tell me just little?
Mister, what I wrote is the process by which plants manufacture carbohydrates from raw
Hmm.
materials using energy from light is called
Uh,
photosynthesis.
very good, very good. Okay, so see here, this is the photosynthesis. You know very well that in plant, there is a chloroplast, and this chloroplast, chloroplast has a green pigment that is called as a chlorophyll. So chlorophyll, has the capability to absorb the sunlight, okay, and convert the light energy into chemical energy. And chemical energy means, like, it makes a chemical compound. And what is the name of this compound? The name of this compound, is glucose. You can see here in the screen. Okay, see, carbon dioxide gas and water react together in the presence of light, and they make glucose and oxygen. Okay, so this is, like, this is the importance of this process, that carbohydrate, okay, that is the food of the plant, is to be made by this process. Did you understand this slide? Anything that you didn't understand, you can ask me, please. Okay, now come to the next slide. Yeah, see here, this is, this is the word equation. See, carbon dioxide gas and water, they react together in the presence of light, and they make glucose and oxygen. Now see here, uh, the what are the sources of carbon dioxide gas, water, okay, and what the products are made, as you can also see in the slides. See, carbon dioxide gas, diffuses into the leaf through the stomata. The process of diffusion. Diffusion means the molecules move from high concentration to the lower concentration. So carbon dioxide gas, that is 0.04% in the environment, it is to be absorbed by the leaf. And in the leaf, there is a small opening that called as the stomata. From stomata, CO2 is absorbed, and water, from where the water is to be obtained, water is to be obtained from the soil. Because you know very well that in the root hair cell there is a large surface area, and due to which, the high amount of water is to be absorbed by the root hair cell. After that, these root hair cells, transport this water, okay, to the xylem vessel. And from xylem vessel, this water is to be transported different parts of the plant, especially it reach to the leaves. Okay, and in leaf, carbon dioxide gas and water, they combine together in the presence of sunlight and make glucose. Now see, glucose here used to make substances needed by the plant. Okay, this is the food of plant, because when so many glucose molecules combine together, they make a starch. Okay, and then starch, is a storing carbohydrates. So here in this word equation, you see that glucose is made, okay, and that is used in the respiration to release energy. Because glucose is going to react with oxygen later on, okay, and then it's going to make energy. And this energy is utilized in the plant for different processes. Now oxygen is also obtained, okay, in this process as a byproduct. Okay, and this oxygen, diffuses out of the leaf through the stomata. and this like this is used in the respiration process. Okay, so this is the word equation and there is an explanation. This is a very good word equation because each and everything is to be clear here. Okay, and what is the source of carbon dioxide gas, what is the source of water, and which compound is made and which byproduct is made and what is the use of that byproduct. Now come to the next slides. Yes, factors affecting the rate of photosynthesis. Yes, what are the factors that affect the rate of photosynthesis? So you know that factors means which particulars, particulars compound, okay, required for this process. So this is like the most important compound, is the like the light. Okay, so light is obtained, okay, from the sun, very well. Okay, so its light intensity is very low, hence the rate of photosynthesis is also very slow. Means if the rate of light is very low, so the process is going to be slower regardless of the carbon dioxide gas concentration and temperature. Carbon dioxide gas is also needed, very well that this carbon dioxide gas is obtained from the atmosphere, and the concentration of this carbon dioxide gas in 0.04% the atmosphere. And temperature, yeah, because the this is enzymatic reaction. So enzymatic reaction, all enzymatic reaction needs a specific temperature to work. Okay, so a specific temperature is also required for this process. Now the another factor is carbon dioxide gas. You see CO2 concentration increase, the rate of photosynthesis also increase. See, as you increase the amount of CO2, okay, so the process of photosynthesis will also be increased. Another factor you can see here, temperature. Photosynthesis requires enzymes to carry out the reaction. As the enzyme have an optimum temperature, photosynthesis also has an optimum temperature. So this is usually about 25 centigrade. Yes, means like if the environment temperature is 25 centigrade, okay, so it is the best temperature. Okay, that is for the working of the enzyme, which has
Wait,
sorry.
Mister, can you go back to the other slide?
Yeah, sure, sure. Yeah, this one. Factors affecting rate of photosynthesis. I've also sent all these slides, to your WhatsApp if you check it.
Okay,
Yeah, yeah. Okay. So this is the
okay,
previous
that
slide.
sounds good.
If you have any question in the previous slide, please.
I didn't get the rate of photosynthesis is limited by the limiting factor.
Okay. First you understand what is the limiting factor. Do you know what is the limiting factor?
No.
Okay. See, limiting factor is the factor which is required, okay, in a minimum quantity to proceed the process of photosynthesis. Okay, so like if the minimum quantity is not available, then process of photosynthesis will not proceed. Okay, so the rate of photosynthesis is limited by the limiting factor. Means when the minimum amount is available, okay, for example carbon dioxide gas, minimum amount is available, then process of photosynthesis will proceed. Okay, but if the minimum amount is not available, this process will not proceed. First thing. Okay, second thing is that when like limited amount, limited quantity of like carbon dioxide gas is available, then this process will start. Okay, and the rate of photosynthesis after that is going to be continue and it's going to be constant. Okay, but without getting the minimum quantity of like carbon dioxide gas, this process will not proceed. So all those factors which are required in a minimum quantity to proceed the reaction, to proceed the process, called as limiting factors. Okay, this is the factor which is least available to the plant. For example, at night light intensity is very low. See, for example, light at night, this light intensity is very low. So what happens, the process of photosynthesis is going to be slowed down. And even like in the night, it is slow but not to be stopped. Why? Because it's still the like the light dark reaction is continue in the plant. Okay, so hence the rate of photosynthesis is also very low. regardless of the carbon dioxide gas concentration and temperature. The same like factors, carbon dioxide gas and temperature. Yes. So see here, carbon dioxide gas, as CO2 concentration increases, the rate of photosynthesis also increases. Means when it's going to just like cross the limit, cross the minimum limit. Okay, then this process is going to be start. But without like getting the minimum quantity, okay, it will not proceed. Yes. Did you understand the limiting factors?
Yes, I do, Mister. Thank you.
Okay. Okay, now come to the next one. Now, is there any problem in this slide? Because I've explained it to you.
No, no, it's easy.
Okay. Okay. After that, I was just explaining you this temperature. Temperature, temperature is also the limiting factor. And the question rises why temperature is necessary. Because, at a good temperature, the enzyme start to work. For example, if you are going to talk about the human beings, okay, in human beings, the optimum temperature for working an enzyme, is 37 centigrade. At 37 centigrade, all our processes like, are going to be proceed in a smooth way. So in the same way, temperature, a good temperature is also required for the plant, and optimum temperature here, is 25 centigrade. Okay, below that 25 centigrade, the rate is going to be very slow down. Okay, and at this temperature, enzyme are going to work. Okay, and at this temperature, enzyme show the best activity means the rate of photosynthesis is very high. Okay, so so the plant photosynthesize slowly as the enzyme have the little kinetic energies. Thus, few enzyme substrate complex are made. At very high temperature, these enzyme denature. Yes, one more thing is here that when we talk about the relationship between the temperature and the activity enzyme. So each and every enzyme has a specific temperature at which it work best. That temperature called as optimum temperature. But whenever like the temperature exceed to the limit, the optimum point, okay, then the enzyme start denaturing. For example, the optimum temperature for human body, is 37 centigrade. If the body temperature become 38 or 39, then we got fever. Okay, and in fever, all our enzymatic reaction are going to be slowed down. That's what, when we are having a temperature or we are having like the fever, we are going to be very very lazy. Okay, and our food is not to be digested. Even we can't stand properly. We can't like perform our job properly. Why? Because enzymatic reaction are going to be slowed down. And after like after the optimum temperature, the time comes, okay, when like the enzyme are going to be denatured or damaged. And once like the enzyme are going to be denatured or damaged, then they cannot take the previous position. That's what like it's very important that the temperature of the enzyme activity is to be maintained. Okay, now second, another factor is light intensity. As the light intensity increases, the rate of photosynthesis also increases. A high light intensity can sometimes lead to the plant heating up above the optimum temperature. This means that the temperature would become the limiting factor, and the rate of photosynthesis would not be increased by further increase in the light intensity. See, like the light intensity is also the limiting factor, because more light or less light, both are the less light mean below the limiting factor, the process will not proceed. But high amount of light increase the temperature. Increase the temperature means the enzyme is going to be danger, enzyme is going to be damaged. Okay, so is that clear? This slide is clear or anything is there? Hello?
No, Mister, I understood it.
You understand? Okay, good. Now come to the next one. Knowledge of limiting factor is useful for farmers as they can reduce the limitation on the rate of photosynthesis due to the factors. Okay, you know that farmers basically are concerned too much about the growth of the plant. Okay, and they will have to maintain the temperature. And you know that each and every, like, plant, has a specific temperature at which its enzyme work best. If you are just going to visit the nursery, plant nursery, you'll see that some plants, are to be kept in the plastic house. And this house we call as a greenhouse. Okay, why they put it into the plastic house? Because these plant needs high temperature, okay, for the working of an enzyme. Because at high temperature, when the enzyme work, then the rate of photosynthesis is going to be very fast, and they grow faster. Okay, because more rate of photosynthesis and more growth of the plant, thus the yields is higher and the growth time is shorter. Farmer can reduce the effect of these limiting factors by placing plant in the greenhouse. See, farmers, they put the some plant in the greenhouse, mean the plastic house. And what is the like the property of the plastic house? Actually, it absorbs the sunlight, okay, and once, the light is to be absorbed, then this greenhouse trap the heat, and thus the plant are always kept at their optimum temperature. In addition, artificial lighting can be used so that the plant can photosynthesize during the night as well, and carbon dioxide gas can be pumped into the greenhouse. So artificially, if you want to just proceed this reaction, okay, or you are going to increase this reaction rate of reaction, then you need to give the artificial light, as well as you can also pump the carbon dioxide gas. Okay, now come to the next one. Identify. Yes, see, identify and explain the limiting factors of photosynthesis in different environmental conditions. See here, sometime they give you the graph, okay, and in the graph, basically, you have to just you have to just tell the point of limiting factors. Okay, see here, limiting factor. You see again, limiting factor is something present in the environment in such short supply that is restrict life processes. Three factors can limit the speed of photosynthesis: light intensity, carbon dioxide gas concentration, and temperature. So here you see there is a graph of all these three factors. See, light intensity and the rate of photosynthesis. See, light intensity. When the plant is going to get the sufficient amount of light, okay, then the rate of photosynthesis, is going to be constant. You can see here. The line become horizontal. It shows that the rate of photosynthesis is become constant. And the second factor you see here, the carbon dioxide gas and concentration. As the amount of CO2 is increased, okay, rate of photosynthesis also increase. Okay, and then the time comes when, we
Wait,
get.
wait, mister.
Sorry, hello.
Mister,
Yes,
can you re-explain the first one? The
this one. Okay. Now see, light intensity. See,
light.
light intensity is to be kept on the x-axis. Yes, it is to be kept on the x-axis, and the rate of photosynthesis is to be kept on the y-axis here. Okay, see, as the light increasing, you can see here, as the light increasing, okay, so the rate of photosynthesis also increases. Okay, and see, at this point, the plant get the sufficient amount of light. Okay, and after that, if the light further increases, that might be dangerous. Okay, that's what, this line become horizontal like this. And this line shows that the light is a limiting factors. Why? Because the plant gets sufficient amount of light, okay, to proceed this reaction. So at this amount or minimum amount of light, this process is going to be constant. That's what, light is considered as the limiting factors. Yes, did you understand?
Yes.
Okay. Now the second factor, is carbon dioxide gas. Again, the same philosophy, that as the CO2 increases, okay, the rate of photosynthesis increases. Okay. And when the CO2 reach to the optimum point, for example, this one, okay, after that, what happens? The rate of photosynthesis is going to be become constant. You can see here. It become constant. Okay. Means this CO2 is also the limiting factors. Okay. Because, if even After this point, even you are increasing the CO2, but see, the rate of photosynthesis is not increasing. Why? Because it get the optimum point. Okay, so in case of light, in case of CO2, there is no, like, the danger point. Okay, even if you are giving the axis quantity. But if you are studying this graph, the last graph, that is like, the temperature and the rate of photosynthesis, you will see that, like, that for the enzyme activity, there is an optimum temperature. Okay, after this optimum temperature, the enzyme, is going to be denatured. And just because of the denaturation of enzyme, the line is going down. The line is going down, meaning the rate of photosynthesis is going down, and the time comes when the rate of photosynthesis is going to be zero. Why? Because all the enzymes are damaged due to the high temperature. Okay, so only this things only can be seen in case of temperature. If that in the beginning, when the temperature increasing, okay, then the rate of photosynthesis also increases. See, the temperature increases, okay, and rate of photosynthesis also increases. And this one, for example, this one, this is, this is the optimum temperature. This is the optimum temperature at which enzyme work best. Okay, if you further increase the temperature horizontally, you can see on the x-axis, if further increasing the temperature, then you see the rate of photosynthesis is going down. Means, like, the enzyme are getting denaturations.
They got denatured, Mister.
Sorry.
The enzymes got denatured.
Yes, enzyme got denatured, okay, and damaged. And once the enzyme denatured or damaged, okay, then the reaction is going to be stopped. You see here, the firstly, like, the this line is going down. It means the rate of photosynthesis is going down, and it reached to the 0 point. You see here, see the cursor here. When it reached to this point, it means the rate of photosynthesis is zero. It means that at this point, all the enzyme are damaged. And once the enzyme are damaged, then it cannot re-get the previous position. Okay, so if a component is in low supply, then the productivity is prevented from reaching the maximum. Okay, so here, only the danger point that comes, okay, in the temperature. But other, like, light intensity and carbon dioxide concentration, there is no danger point if even you are increasing the carbon dioxide gas or light density. But you know the rate of photosynthesis neither increasing nor decreasing, means it fixed at the a specific level. Okay, but in case of temperature, okay, if the temperature, reach to the optimum point, and after that you are further increasing the temperature, then it gets the danger point. Okay, and at this temperature, like, high temperature, all enzyme are going to be damaged, and the rate of photosynthesis is going to be zero. Why? Because like, enzyme are not working. Now see one more thing here about the sunlight. Light energy is vital to the process of photosynthesis. It is severely limiting at times of partial light condition, for example dawn or dusk. As the light intensity increases, the rate of photosynthesis will increase until the plant is photosynthesizing as fast as it can. At this point, even if light becomes brighter, the plant cannot photosynthesize any faster. Okay, so see here, like, light intensity is there. See a point, say a point. A point means, like, as you increasing the light, the rate of photosynthesis also increasing. Okay, and from B to C, you will see that there is no changes in the rate of photosynthesis. Means the light intensity is increasing horizontally, but rate of photosynthesis become constant. Okay, B to C, the rate of photosynthesis is become constant. Is that clear or not? Limiting factor.
It is the same.
Okay, now come to the next one. uh, yes, identify, explain the same topic. Over the first part of the curve between A and B, light is a limiting factor. The plant is limited in how fast it can photosynthesize because it does not have enough light. Now, between B to C, light is not a limiting factor. B to C, light is not a limiting factor, even if more light is shown on the plant. Okay, means B to C, more light is coming, so that's what, like, it's not a, in this way it is not a limiting factor. But once the time comes, then it's going to be become constant. That means, like, it's a limiting factor. Now, carbon dioxide, if in photosynthesis CO2 is the key limiting factor, the usual atmospheric level of CO2 is 0.03 percent. You'll see that the amount of, or the percentage of CO2, is mentioned 0.03%, or in some books it's mentioned like, 0.034 4%. Okay, so just a little difference, in the variation of the books. So in perfect condition of water availability, light and temperature, this low CO2 level holds back the photosynthetic potential. The more CO2 a plant is given, the faster it can photosynthesize up to a point. Okay, but then the maximum is reached. So, as you increase the CO2, the rate of photosynthesis also increases, but after that, it goes to the uh, like, some particular limit, and then the rate of photosynthesis is going to be constant. Temperature, the chemical reaction of photosynthesis can only take place very slowly at low temperature. So the plant can photosynthesize faster on a warm day than on a cold day. Now here you see there are some experiment, because, we are studying here not only, like, paper 2, 4, but also paper 6. Okay, so some experimental questions are also given, in paper 6. Even some questions, are given in paper 4. Okay, these are the investigating questions. So see, investigation, getting the need for chlorophyll, light, and CO2, carbon dioxide gas. If you are just going to study, like, this equipment, you see, we take a beaker, okay, and we put, like, the like, water, and then put a leaf. Okay, so leaf, and then we are going to start to heat this water. So this leaf, is in the boiling, is in the boiled water. So what would happen, when you just, like, put the leaf in the boiled water? So there is a breakdown of cell wall, okay, in the cell of leaf. This is the first step. Okay, okay, second step, what is the second step? In the second step, you keep this leaf in the alcohol. And what is the purpose to keep the leaf in the alcohol? To remove the chlorophyll from the leaf. Okay, so if you keep the leaf, okay, in alcohol, ethanol, alcohol, okay, then, like, the chlorophyll, that is the green pigment, is to be removed, is to be removed. Okay, and okay, after that, number 3, in the third step, what's going on? Leaf washed in water. After that, the leaf is to be washed in water, and then, like, the next step.
Mister,
Yes.
it's washed in the same hot water?
No, no, the we wash it, okay, in the cold water. Okay, so that the leaf
Okay.
become soften. Okay, then leaf placed on the white tiles and covered in iodine solution. Okay, after that, see, in the first step you remove the cell wall, okay, in the boiled water. In the second step, you remove the chlorophyll, putting the leaf in the alcohol. Okay, in the third step, you put the leaf, okay, in the cold water, so that the leaf becomes soft. Okay, and then after that, you put the leaf on the white tiles, okay, and put some drops of iodine. Okay, and you know that, if like, if there is a presence of a starch, okay, if there is a starch is in the leaf, then the color of iodine, that is normal reddish brown, turns to blue black. If it turns to blue black, it means that the starch is found in the leaf. See, reason, iodine will change the color in the presence of starch. And what color change? See, the normal color of iodine is brownish red. And when if there is a star, then it turns to blue black. You can see here in the last, the last step. So areas that contain chlorophyll will turn blue black as stars will be present there. Is it clear?
Yes, Mr. It's clear.
So actually, this experiment based on the detection of starch, okay, in the leaf. Now come to
Okay.
the now come to another experiment. Another experiment. See here. Yes, another experiment. Investigate and describe the effect of varying light intensity, carbon dioxide gas concentration, and light intensity, and then, another thing, that is temperature, on the rate of photosynthesis. See here. Actually, this diagram mostly given in paper 6 and sometime also in paper 4. Okay, see what we are doing here. We take the beaker, okay, and put the aquatic plant in it, and then invert the funnel on it, and also, invert the test tube, okay, and we give each and every requirement to the this plant. Okay, means this plant needs water. It is in the water. Okay, it needs carbon dioxide gas. How carbon dioxide gas is to be given? We put some compound, sodium hydrogen carbonate in water. This sodium hydrogen carbonate gives carbon dioxide gas to this plant. So this plant is getting carbon dioxide gas, water. Also, this plant is getting light from this lamp. Okay, and chlorophyll is already there in the plant. So that when CO2 and water react together, okay, react together in the presence of a specific amount of light, then the process of photosynthesis will proceed. And in process of photosynthesis, you know very well that oxygen is evolved. So see, oxygen is released, and the bubble you can see here. See, if you see more bubbles, it means the rate of photosynthesis is very high. If you see less bubbles, so photosynthesis rate of photosynthesis is going to be very slow. How can you just increase and decrease the rate of photosynthesis? See, if you keep the lamp just near to this plant, okay, the rate of photosynthesis will increase, and you will see more bubbles. If you keep this lamp little away from the plant, so you will see that the rate of photosynthesis is going to be very slow down, and the bubbles are going to be less. Okay, in the same way, if you are going to add more sodium hydrogen carbonate, okay, so more CO2 would be there, and more CO2 means rate of photosynthesis is going to be very high. For example, in 1 minute, in 1 minute, if you just count 5 bubbles, okay, of oxygen, if you are going to add sodium carbonate in more quantity, so the bubbles are going to be more, which means that rate of photosynthesis is increasing. In the same way, if you keep the lamp near to the plant, you will see the number of bubble will increase, which means that the rate of photosynthesis is directly proportional to the light intensity. More light, more bubble mean more rate of photosynthesis. More CO2, more sodium hydrogen carbonate means more carbon dioxide gas and more rate of photosynthesis. Is it clear? Okay.
Yes,
Now
Mr.
come.
It's clear.
Okay. Now come to the next experiment. Yeah, this is about the same. You see here. Okay, factor we investigated. Hot plate to change temperature. Okay, the same one, same philosophy is there. Okay, here also the same philosophy, because you see, uh, we are just going to keep the lamp position near, okay, or little away, and we will see the rate of photosynthesis is going to be changed. Okay, and one more thing is that see, we put the thermometer here. Why we put the thermometer here? See, if you are just keeping this lamp very near to the plant, so may, the temperature is going to be increased very fastly. Okay, then this temperature is to be measured by the thermometer, because if the temperature is going to be more, then the rate of photosynthesis is going to be stopped. Okay, so we just check it, if the temperature increasing very fast, then we keep this lamp little away from the this plant. Okay, now our another topic, the structure of leaf. Structure of leaf, you see here, if you are going to study the leaf structure, okay, so you'll see the uppermost layer is the waxy layer, is a waxy layer. This is we called as a cuticle. And what is the function of cuticle? The function of cuticle is to prevent the transpiration, okay, or is to stop the loss of water. And after that, another cellular layer is there down the cuticle. This is called as upper epidermis. And what is the function of upper epidermis? It reflect the rays of light. It's reflect the rays of light. Okay, after that, the second cellular layer, that is palisade mesophyll tissues. And these palisade mesophyll tissues, have the high amount of chloroplast. High number of chloroplasts are there, and rate of photosynthesis is going to be very high there. And the third cellular layer, is fungi mesophyll, which also have the chloroplast. So these cells are also responsible for the process of photosynthesis. And also they have the air spaces for the for like the for the transportation of the gases. After that, the lower epidermis is there. In lower epidermis, you will see that the guard cells are there. Between the two guard cells, there is an opening. This opening called as the stomata, where the exchange of gases take place and the transpiration take place. Now see the guard cells. These guard cells, when they take up too much water, okay, then they become turgid and the stomata opens, okay. And when they lose the water, so they shrink. These two guard cells shrink and the stomata close. That's what in the night time, mostly the stomata are closed. So the rate of photosynthesis is going to be very slow down. Is that clear? This diagram you understand or not?
Yes, Mister, but I
Okay.
have a
Okay.
weak.
Okay. One more thing is there, that is vascular bundle, xylem vessel and phloem vessel. Xylem vessel, actually, there are two type of tubular system in the plant. One is the xylem, that is responsible to transport water and minerals. Another is phloem. Phloem, responsible to transport amino acid and sucrose. So this is the function of xylem vessel and phloem vessel. And in the given notes, you can also study the function of each and every part. You see, waxy cuticle, upper epidermis, palisade, spongy mesophyll, lower epidermis, and guard cells. Okay, then stomata is there, very well. Okay, and then vascular bundle means xylem and phloem. So we have studied, like, the structure of the leaf and their functions. Okay, so in notes you can also study. Now the meeting is about to be finished. Okay, I'm going to again, just like, again I'm going to send you the link. Okay, then you can just like, continue the lecture. Okay,
Okay,
so
Mister, so we
yeah,
can